Good afternoon, and thank you for this really magnificent introduction. I don't deserve any of this. I'm very glad to be here. I really want to thank the organizing committee for extending the invitation for me. And in the next 30 minutes, I will share with you my experience trying to figure out the complexity of coronary artery disease, what we've learned, and how we're going to carry this forward. My lecture really comes at a good time after Professor Morris this morning, as I will share some of the difficulties that we faced being or studying complex diseases on populations that have not been exhausstly studied by the international community, and actually we have started to change that. So I'll start by this. Tomorrow I'll be going to my hometown. We just had a huge snowstorm. So I come from an area not very far from here. I thought I'll share this sentimental moment with you. And back to reality. So coronary artery disease, we know for a fact that from epidemiological studies and other clinical studies that the heritability of coronary artery disease is anywhere between 40 and 60%. But most of what we know today, with all the genetic studies, genomic studies, and sequencing studies as well, we're still hovering around the 10% heritability that is known heritability to us today. And same goes for diabetes, maybe a little more. But that's where we are. And the vast majority of heritability is still unexplained. I used to teach a course called Environmental Genetics, and the first lecture, the first slide, I would show my students this by a famous lipidologist who would say, well, the first part of this may be explained by monogenic components of cardiovascular disease. But the second part is that, yes, some people do live for a long time despite really how little they pay attention to the environment. And all of this is because, yes, CAD is really a complexity of interactions. There are multiple factors that actually influence this particular disease. And this was the approach that I took when I first started working on coronary artery disease, in a way, in a naive way, but based on what we currently knew at the time. And in 2007, actually late 2006, I started on an ambitious project funded by the European Commission to look at novel factors, me and a group of collaborators, of course, to look at novel factors that we could actually classify as biomarkers or genetic markers, looking at the genome, but also looking at the transcriptome and the proteome. So we were very ambitious in our proposal. And we thought we'll generate these markers, and these would be actually used to predict coronary artery disease. So Lebanon is a relatively small country with a lot of really important medical care. So it was very easy for us to access the clinical setting and to recruit patients who actually simply were undergoing cardiac catheterization. So we would approach the point of care person, and we will contact the patient. If the patient agrees, to sign a consent form, and the patient will participate. So people don't believe me when I show the slide, is that the overwhelming majority of the patients who actually underwent and still undergo coronary angiography are basically doing it as an elective procedure or part of their clinical workup, which is, I thought, was astonishing. Yes, a smaller portion, they were referred to cardiac cath because of unstable angina, and a smaller majority for MI. Of course, if you are an epidemiologist or a astute by statistician, you're going to tell me, well, you're going to be missing a lot of the important ones. Unfortunately, these are the ones that actually succumb to their MI and are not part of the study. So that's one part of the missing heritability that we'll miss forever. So, and we wanted to collect information, and those of you who actually are in this field of collecting clinical information on patients, you have a dilemma how much to collect. Of course, the more the merrier, but unfortunately, you know, you cannot tell a patient to fill a questionnaire that is a 50-page questionnaire. You have to limit it, you have to make it as easy as possible, and as objective as possible. So we limit ourselves, and we've collected successfully about 80 parameters on these patients. In total, to date, we touched the 10,000 6 or 8 months ago, and the majority are males, as expected. And you know, as a quick summary, if you wish, of the clinical conditions of these patients, yes, being a female, you tend to develop coronary artery disease at a later age, so you're protected somewhat. But also HDL in our population is a very protective factor. So I will point these two out quickly. That unfortunately, in the Lebanese population, HDL tends to be on the lower side. And then, so if people with high HDL tend to be more protected for coronary artery disease than not. And I cannot really pass without talking about consanguinity. You know, it's also prominent in Lebanon, maybe a bit less prominent than in Saudi Arabia, but in certain villages it reaches 40%. And what I would like to point here is the fact that when you combine history of disease and consanguinity, the risks significantly increase. And I will let's park this thought until the end of the lecture, because I'll use this in our search for the missing heritability in this case. But it's impressive to see the significant increase when you combine family history and consanguinity. Of course, the recessive alleles with high impact are likely to be the cause of this. So once we collected these samples and we wanted to do the first GWAS analysis on them, we did some PCA, just you know, structure to see where we place these samples. And we were not surprised. So these were the CEF samples, or the Europeans, and these were our FGen card samples. So this is our cohort. We called it FGen card. And then when you spread the PCA out with the Europeans, so these are our samples on top of the Greeks, and then the Italians, and then further the Spaniards, and these would be the North Europeans. So really, as expected, I've been doing this work for quite some time, and this was not surprising to us. And then we proceeded with our first GWAS analysis on phenotype. So we had the stenosis level on each patient. So we said, okay, we're gonna do stenosis versus non-stenosis, and we in this study we did 50% stenosis or more or less as a control, and of course we had several GWAS significant SNPs. And yes, it's all good. You know, we had 19 that reached genome-wide significance, and we had a couple with specific addition to them, so severe stenosis and dyslipidemia. So all of this is good. And then factor 1, which was really the most common SNP that has been, or the locus that has been associated with CAD in several populations. Yes, it was there. It was not one of the best ones we've had, but it was up there. And then I added the slide for Professor Morris here is, yes, we did the type 2 diabetes as well, and the usual suspects actually came, and the majority of them are in the population. So we don't expect much difference in this, and we can talk about this a bit later. But then you try to publish the work, and then in 2007, factor 1 was actually already published, by McPherson, and then so you end up, yes, publishing the work, but 4 years later, and that's because really you're at a 4-year disadvantage here. But the fact is, people love the work because yes, it's from a population that has not been, that has not been looked before, and then it's still important. So it's important to do this work. But then at that time, when I was really realizing that we are lagging behind, and we're not going to find anything new. We expected to find something new in our population, and we're not, because of these common variants that actually are the ones that are going to be discovered initially. So I was reading this article by a colleague of mine and who became a friend, Martin Farrell, who is one of the most prominent scientists in coronary artery disease statistics. He had published this review article that I read, and in this article he talks about how important to actually have the larger data sets, the easier for us to actually get to these steps and to be able to get more of the rarer variants, etc. And I thought to myself, well, why not join a large cohort? And I think, that's the way to go. And that's what we did. So we joined this cohort, which is now called the cardiogram plus C4D. And then, yes, as you can imagine, after that, we were part of this consortium, and we were seeing our work and our population being represented now in these large consortium studies. So the first one that came out in 2013, it adds to the wealth of already important information by adding new loci. But the catch here is that the number of samples was more than 60,000 samples. And of course, 15 new risk alleles. Yes, it's not there yet, considering the whopping number of 63,000. The problem with large cohort, as we heard this morning, is that what is your phenotype? Are we looking at the same phenotype across the entire cohort? When you start mixing cohorts, smaller cohorts together, you start losing this definition of the phenotype. And I think we are seeing some of that. And that's part of the problem. It's not the only problem. And the total number of the confirmed CAD loci has significantly increased. And also, with all the outcomes, yes, that we have, all of them with either blood pressure or inflammation. But what is important in this is that we are still at about 10% heritability. It's impressive, but it's really not where we want to be. So then, again, 2 years ago, now the number has increased as more people joined the cohort, 185,000 cases. If you can see that, and several million SNPs, so close to 7 million SNPs now. And there's a fine print which you cannot see here, which I will highlight to you, is leveraging phased haplotypes from the 1000 genome actually made a huge difference for basically reanalyzing what we had and addition. So here another 10 loci have been added now to the wealth of information that we have on coronary heart disease. All this is great. But then the remaining heritability is still a problem. There is still a problem with actually pushing the limits, pushing the envelope further. Yes, we knew this before we started this, that it's substantial clinical heterogeneity of this. We have not really put our finger on this as we should have, in my opinion. And a lot of these variants actually that do not reach genome-wide significance are still important that we have not looked at carefully, that we need to look at carefully. And of course, the genetic architecture of the genome is far more complex than we had initially thought. So all of these have been addressed to a certain extent, in my opinion, by some of the studies that we were part of in this one, for example. Not this one, I'm sorry. This I give an example for a fascinating study that has been published also last year. It identifies 65 new breast cancer loci from previously looked at GWAS analysis. But what they did is the fact that they looked at distal regulatory elements. Actually, a lot of these SNPs that were looked at, not significant or barely significant, actually, they were proxies for the significant SNPs that were not actually in the exome or the sequence. And I think it was Mark who asked this question yesterday. There's a strong overlap between the candidate target genes and the somatic driver genes as well. So we are understanding a lot more of this architecture of the genome, and as we learn more about these, actually, we're going to be able to tackle this heritability problem much better. So our work, our recent study, is basically now leveraging all this information in, and of course, adding the UK Biobank data addition, which has been magnificent. 9 new loci have been identified, and by performing FDR-based analysis, false discovery rate analysis approach for these suggestive evidence, we actually were able to identify additional loci, now 13, and 12 of these loci that have been identified actually were part of the suggestive, so 5% FDR, so which was really fascinating. And I think there's a lot more to be done on this. So these, as I said, 12 of the 13 that were discovered newly really were either themselves as SNPs that actually were in the 5% false discovery rate, or they were at a linkage or a correlation or by proxy with more than 80%. So basically, we added a little bit to the heritability component that was missing, okay, an addition of 1%. And then before we actually started, the information was, 8.53. Now, these calculations actually, they vary from one tool to another. But still, there's a lot of missing heritability. And yes, we may argue, and I as a geneticist would argue that there should be no reason why SNPs or variants with high penetrance and low frequency should not explain a big part of this heritability. I truly believe that. But I also believe that endophenotype is actually complicating our story in a significant way. And maybe the answer would be in pathways. And to get to pathways, it's far more complicated. But we have to make it somehow. And what I propose, and this is where I'm going with my work right now, is the following. So yes, there are structural effects of the genome, methylation, epigenetics, etc. And we are now looking at this by looking at the structural variants of the genome more. But more importantly, let's look at the multifactorial impact of CAD. And this is what I used to teach my students is, yes, there are multiple pathways leading to disease. So at the end of the day, yes, we'll get to stenosis, but how do we get to stenosis may be very, very different from one patient to another. And unless we really classify those patients, we have, maybe we're going to have 20, 50, 100 phenotypes for CAD. Why not? But let's look at them as individual endophenotypes. And of course, somebody asked earlier today about the microbiome and metabolomic interaction. And of course, we are seeing this with our diabetes study. We were fascinated to actually discover a compound that is secreted by a gut bacteria, which actually has a huge effect on insulin secretion in humans. It's fascinating. So all of this has to be addressed in a comprehensive manner in order for us to push the envelope a bit further. So moving forward, my colleagues and I were proposing that, let's look at patterns or clusters of phenotype and see if we can use this clustering of phenotype that we can associate these to specific pathways. And then once we have these pathways, we can look at underlying mechanisms. And these underlying mechanisms could be as candidate genes that we look at, or a set of candidates, and really conduct the analysis now based on these networks and on these pathways. And the way we propose to do this, and we have started to do, is that we take our cohort and we look at clinical variables and we try to classify them. The problem is we are seeing a lot of these small groups, okay? They all, you could see huge blocks, but you could see also large number with small distinctive subgroups. So, we're gonna do a lot more on this. If you look at this, for example, we are starting to see this. If you look at these, for example, the red cluster here is you have age over 60 with type 2 diabetes and with cardiovascular disease. Okay, blue, this one here is obese. They have high LDL, hypertension, and CAD. So we are really trying to do this, and imagine that each group here will be treated differently and will do the analysis. Now, you may argue the fact that, yeah, we're going to lose significance from having the number, low number, but we may actually gain it from having a better phenotype with a more impact on that particular variant. So it's a start, but this is where we're going with this. Add to that, remember the first slide that I showed you about family history and consanguinity. So it turns out that when I looked at our population, and when really looked at the consanguinity and family history of the disease, there's a 10-year difference between the age of onset among this group and that group here, or this group here, which do not have risk factors of disease and what have you. So this group actually could be one of those clusters that we're going to focus on and work towards. So our population gives us some ideas or some clustering already that we don't need to go through hoops to actually get there. And I can even take this further. So this group here has family history of first degree and then consanguinity of the first degree. So you can even subgroup those and actually look at those separately. Yes, it's a complex disease. Looking at it from that perspective, you may favor alleles with large impact, but we need to get these out of the way as well. And then finally, I mentioned metabolome and the microbiome. Yes, we are doing this as well. And basically, what we're doing with this is the fact that we started on a subset of our samples. We analyzed by NMR the metabolome on these. You clean them, and then you prep them, and then you do the same set with a genome. You filter the SNPs, and then you end up having two sets here to compare. You do the MWAS, metabolome-wide association study, and the GWAS, and you compare the two. And we are really getting a lot of interesting findings as we speak. Of course, it's a small number, but we are getting there. And I think it's the combination of looking at these endophenotypes, looking at the structure of the genome further, and adding the metabolome will get us there. So the moral of the story is, yes, we are in the era of omics, but we have to really harness omics with the background information that we have, heavily clinical information and structural information from the genome. I will stop there and say thank you very much.